Hello to those who've already joined. We're just going to wait a couple of minutes just to make sure that everyone can get in the call before going. So hold tight and we'll be with you soon. Right, we're 2 minutes past the hour. Are you all right if I get going, Tim?
Yeah.
Fabulous. Welcome to the PCSIG webinar. For anyone who is eagle-eyed, you will be expecting Neil Spooner, chair of the PCSIG here. As you can see, I am not Neil. Neil is sunning himself on holiday, so I'm afraid you've got me instead. Jenny Royal, who is a different board member of the PCSIG. We're here today to listen to a talk by Tim, but before we get going, it is my pleasure to say a huge thank you to all our partners and sponsors. Without our partners and sponsors, we couldn't do the range of activities that we do. So thank you. Everybody is on here and we very much appreciate it. Another little bit of housekeeping before we get going, and I leave you in Tim's capable hands. The Q&A, please use the chat function to submit questions. We will be ending with time at the end to go through a few of the question and answers. If we are inundated, we will do our best to pick out as many as we can. So please don't hold back. We'll be trying to ask them at the end. Which means I am here to and have the pleasure to introduce Tim Woolley, who is Chief Scientific Officer of Inuvi Diagnostics and also Editor-in-Chief of the Journal of Patient Centricity that you will hear more of later on. Tim is here to talk about the UK recommendations on the validation and adoption of capillary blood testing within labs and the consensus opinion. So without further ado, over to you, Tim.
Hi, good afternoon, everyone, and thank you for joining today. Thank you, Jenny, for that intro. That's taken away the first slide I was going to talk about, so that's removed any buffer I've got. So if we could, I'm just going to quickly go over why we've done this, why the PaCTS group put in place or looked at the recommendations. A lot of feedback that we'd had initially was that people didn't really know how to start the process of validating capillary blood. So we put together this set of recommendations, and it is only a working document. So what I wanted to do was just give you a bit of a background to capillary testing and where it's come from, just a rough background. So capillary blood's been in place now for decades and decades. You can go all the way back to probably the 1950s, 1960s with the Guthrie cards. It's been used on the wards for a long time, pediatrics. It's been used for needle-phobic patients. It's been used for obviously point of care in sharp controlled areas like prisons. But most of that work is being done by the professionals collecting the blood. And then there's been a little bit of a difference with things like the STD or STI outreaches, where you can go online and you can order your kit, a urine, a swab, or a blood test, and you can send it back to the lab for testing. So that's been going on for probably more than a decade now. So that kind of started the ball rolling with this. And then a few years ago, probably around about 10 years ago, some of the online providers started offering capillary blood testing via the postal network. And then during COVID, the UK managed to test, I think, 4 or 5 million people using self-collected capillary blood for COVID antibodies. And certainly my lab was one of those labs that were fortunate enough to do some of that work, and we learned a huge amount. At one point, we were doing about 9,000 samples a day. So capillary blood is not new by any means. What's different here is that this is now being used for lots of different tests, not just a few that's been out there for quite a few years. And you can go online, like I say, you can order a whole vast array, and the validity of those sample results were being questioned because there was very little being published. Although over the years there has been quite a few papers being put together, a lot of that didn't include things like postage, didn't include sample stability. The comparability work was being done there and then with venous work. So we so some of the problems that arise from that is that sorry, go back. So some of the problems that arise from that is that people want to know whether or not their tests are valid. And there's been a lot of work being published this year on a whole different array of different comparability studies. And so the future is actually now. We're doing this right now. Most of this work is being done somewhere in the world on capillary blood testing, being sent back to the lab. And you can look at any of those. Obviously, the infectious disease we've talked about, there's been papers being published on things like cancer markers and liver function and renal function and lipids. Some of the work's been already been done on response to treatment and therapeutic drug monitoring. That's been done for quite a long time as well. So these things are already happening. The problem that we have is there's not a lot of published evidence out there. And also the most important thing, which is part of the problems, is that there's no agreed way of doing the validation process. So this is quite scary for a lot of labs, if you like. So let's quickly just cover the problems. Collection devices. Until quite recently, finger prick has been the main way of getting a capillary sample. When I was younger, they used to do it from the earlobe. I've even seen people take it from the nasal area there, which is frighteningly painful. But recently, there's been some of the upper arm devices have come out, which have made it almost painless. The sample quality is really, really high. The amount of samples really, really good. And we're on to probably now the 3rd generation of those devices. Some of them are coming out now that can do several tubes. So the problems that have been around the collection and the hemolysis and things like that kind of gone away to a point. The validation protocols we'll touch on in a second. But there's a growing number of stability and comparability studies, and there's been a couple, 3 or 4 out in the last month or so that have looked at things like lipids, creatinine. I think there's been a couple on LFTs. So, and they're looking at different areas, and these have included postal samples. So that's kind of really, really, really gold standard. But the problems are around very little automation in the lab. So it's quite time consuming when you get into the lab. It's easy to do if you do small amounts of work, but when you start doing large amounts of work, unless you've got a really, really high-end bit of kit, it's quite difficult to get this automated. Likewise, the economics, which is cheaper, where is it appropriate, is also up to question. And then the kind of feedback that people had was that there was limited knowledge of how to do the validation protocol. So why validation and why verification? So for people who aren't in the lab, the difference between validation and verification. So if a kit insert for an assay has serum listed on it, that will mean venous serum. It won't mean capillary. If it's got capillary listed as a matrices on that IFU, then it's CE mark for use for capillary. If it's got that matrices listed, the lab will do a verification. And what you do is you verify that your performance in the lab is as good as or better than the manufacturer's claims. If the matrices you're using is not specifically listed on that IFU, you need to do a validation. And validations can vary in complexity. We're using, when we first started, we've used the CSLI EP35 as a guide. And that says that you can do a validation by comparability. So if the matrices are very alike, for instance, in this case, whole blood, then you can do a validation by comparability. More recently, Roche have released some of the assays now that have got capillary blood listed as an approved matrices, so you'll just do a verification. But the MHRA, obviously in the UK, have specifically said if the capillary is not listed on the IFU, then you need to do a validation. So the PaCTS group looked at this and said, well, it'd be great if we had a consensus opinion of the right direction to take for labs to do validation, and that's kind of what we did. So this is an outline of the flow diagram from the paper. Excuse me, and it lists lots of different things that a lab should do. First and foremost is you start off with a change control, and that change control is there to capture all the information about that validation. And the most important thing here is that you ask whether or not there's any point to it for the patient. Is it going to improve that patient pathway? If it's not going to improve anything, then it's probably not really appropriate. But start off by defining the patient cohort. Is it a well patient cohort, or is it a screening cohort? Whatever it is, identify the patients you're going to be wanting to test this on, and get those individuals involved early on. Because if you do all the work and then go back to the patient and say, well, we're going to do this, and they go, I don't want to use it, then it's kind of been pointless. And there have been papers that have shown that a number of patient cohorts love this, but there's been one or two that have said actually they don't really want to do it. They prefer to come into the clinic because they get blood taken a lot, they need other things, they need medication, they want to speak to the doctor. And if it's not appropriate for that group, then you've wasted your time. So first of all, define your patient cohort and speak to that patient cohort. Then you need to look at a risk register, look at your risk assessment. What's the harm that could come to the patient if you do this? Not only during the testing phase, but also once you've done the testing and you roll it out. If, for instance, you're replacing monitoring visits, they come into a GP and have some blood taken, or an outpatient, and there's no downstream negative effect of a failed sample, then it might be relevant for that patient group. If, however, if they get an incorrect result, for instance, from an insufficient or hemolyzed, or they forgot to take it, if that has a massive effect on the downstream patient care, capillary probably may not be appropriate. So you look at all those things, look at the risk to the patient, and document it, and also write down your thought process. Then you look at, we'll come on to the comparability side in a second, because that's kind of where we're going to be focusing on. But the other things you need to think about is IFUs. So how do you teach the patients to take the blood sample? Is it training in person initially when they come to a clinic? Is it online content? Is it a hard copy? Do they need a reminder? Is it the patient's going to take it, or is it the carer? So that IFU needs to be based for that patient group that you're looking at. And different patient groups will need different IFUs. Likewise, you need to document the acceptance and rejection criteria for the patient group as well. So if you take, for instance, a particular patient group that never has blood taken because you can't get blood off them, and you have a failure rate of 30%, then actually that's 7% of people that have had blood done that have never managed to have blood done before. So that's probably a bonus. Whereas if it's something where you get 99.9% of people get blood samples all the time via venous, and you start bringing in a 15% failure rate, it's potentially that capillary is not the right appropriate method for testing. So we'll come back to this a few times, but if we move on to the next slide, which is starting to look at the change control and the risk assessment. Now, I've kind of gone over this already, but this is where you list all the variables that you're going to be wanting this service to provide. So if it's self-collection, or is it professional collection for capillary? Is it going to be sent back in the post? Are the patients going to be dropping it off in a GP surgery? How long will that sample take to get back? If it's dropped off at a GP surgery, then it's probably going to get picked up by the hospital transport and back into the lab within the same sort of time as a venous sample would. In which case, you probably won't need to do as much stability work, just comparability. If you're going to be putting these samples into the post, it's how long is that sample going to be in there, and what sort of temperatures is that sample going to come into contact with. So Look at the method of collection. Finger pricks really quite cheap. Upper arm technology is more expensive, but you get a better quality sample. It's easier to use. They often come in packs that are pre-made. They're CE marked for self-collection. There's a whole range of things that are kind of the benefits of the upper arm technology. And then you need to look at ethical approval. If you're going to be doing this on a particular patient cohort and you need to get that patient cohort included, then you're probably going to need to have samples from those people to do your capillary validation. So that might take a little bit of time, and that will need to be done as well. And then for the risk assessment, as we've said, identify the patient cohort, get them involved early on, and this is patient-centric testing. So the quicker you can get patients involved, the better the outcome will be. You'll start to see if things start to go wrong, you might need to rethink the whole process. This is also going to be looking at whole process validation, as we've said. The capillary sample is the pre-analytics are out of the lab's hand, so you really need to get people involved early on to find out how they're going to collect those samples. What you don't want to do is do a fantastic validation to find out that you can't get the right amount of sample, or it's hemolyzed, or people are labeling the tubes wrong. So look at the whole thing, including the transport back to the lab. And you design the testing protocol, the validation protocol, to try and include all of the risks and mitigate these. So you literally are listing all the things that could go wrong, and you're adding those into your validation. So it's got to be a real world validation. And then, of course, add it to your risk register, the lab risk register, and that'll need to be revisited over a periodic time as different tests come online. So the validation plan, again, it kind of splits into three areas, and there are a lot of crossover. But first of all, you need to include, as I've said before, the patient. Ideally, the patient cohort, discuss what you want out of this project with the patient and what they want out of it. Is it what kind of methodology would they be useful? Have a few tests, show them things, get their buy-in before you go anywhere. Because if you can't get those buy-in, it's not going to go very far. For the laboratory side, you need to things like find out what reasons of failure you're going to have. That also comes into the service, but we're going to see more hemolysis, more insufficient. How we're going to deal with that? Have your acceptance criteria listed. So we recommended that people go to the EFLM total allowable error, because that's an international source of total allowable error. It's fairly standard and very robust, but you might want to have something different. But that's kind of a first place to go, is find out when you get your results, your comparability results, what are you going to accept as a difference? And then what statistical methodology you're going to use for that. Again, we've recommended a couple, and we'll come on to it near the end. But it's things like Deming regression, Passing-Bablock, and Bland-Altman, using the EFLM total allowable error as your acceptance criteria. And then for the service, speak to the consultants in charge of the service, find out what the errors could have effect on the patient. So if, again, we've said before, if the failures are too high, is that going to have a knock-on effect? It might save money at the GP clinic or the outpatient. The lab might be able to handle the costs, but if a patient can't get the results, are they going to go to A&E? Are you going to get a lot of people suddenly going to A&E for blood tests because they can't have medication, for instance? So look at the worst case scenario, and managing people's expectations on this is obviously the right thing to do. If you say that your failure rate is going to be 5% and actually it's more likely to be 10%, then tell them the truth. Tell them it's going to be 10%. And if they accept the worst case scenario, over time it will get better. There's been evidence shown that with upper arm technology, the failure rates can be, from different reasons, can be anything from sort of like 20 down to about 5%. But when you start to get people getting used to the technology, that can drop to about 2% failure rate. Now, when you look at venous failure rates, that is around about 1%. So it's not too far off. But again, it'll depend on a number of things in the validation plan. If you're looking at doing 20 tests for a capillary sample, your failure rate is probably going to be higher than if you're doing 1 or 2. So you have to build that in. We're now coming on to the more of the lab part. And what's important here is that once you've identified the patient cohort, that comes back into the comparability and stability. What you want to do is get a number of samples over the, ideally over the limited detection for the assay. But if you can't do that, it's really important that you cover off the expected level of results you're going to get from the patient cohort you're testing. If you've managed to get hold of samples from the lab staff, for instance, then they're all normal. That really isn't applicable to a group of people that may have different thyroid diseases. So you're going to really have to try and stretch and get as many of your patient cohort involved with the study as possible. And then we've said that the EP35 recommends 40 paired samples. Now, we've also included the 40 pair samples, and in the EP35 it also says you can go down to 20 with justification. But the most important thing here is that you get across that that result range that you want. I did a study not so long ago on a marker, and we managed to get within 22 people, we managed to get everything from the lower level of detection all the way up to the upper level of detection, all the way along the linear line. And we added more data to it later, but we'd covered off everything. Now that's quite rare. You won't do that with things like thyroid or maybe some of the liver enzymes, things like that, where there's a very wide limited detection. But we managed to do it for it was LPA in this case. We've just done another study on BMP, and we can't get any high BMP results. So there's a lot of work to be done there, getting the right patients and getting the samples from those patients. We've said that 40 paired samples is appropriate. When you're doing the assay, when you're doing the verification, the validation protocol, do you need to include sample stability? Again, if you're dropping the samples off within a clinic, probably not. But if you are going to include, if you are going to post those samples, then try and build that into the validation. Look at the kit insert. Most of the kit inserts won't include whole blood stability. Most of them only have stability for serum. It'll say 3 days at 4 degrees or something like that. So you will have to build in whole blood stability. And you'll also have to consider the transport conditions. Is it going to be like 30 degrees at the moment? If it is, then you might want to build that in. Fortunately, Britain is usually pretty cold. It's fantastic for posting pathology samples. The last 2 or 3 weeks have proven that that's changing. Do you need to consider that? So you might not be able to get lots of capillary samples from the same people over and over. So the way that we've recommended is that you get a venous sample and a capillary sample taken at the same time. Ideally, you'd get another capillary sample and test them at that 0.0, and then do them several days later if you need to do postage, or if you are only going to be doing them within the IFU time frame and stability, then you can do them there and there. But you must also always consider what transport time and conditions that those samples are going to be transported at. When you do the testing on the machine, we've included things like rather than use the capillary samples for precision, use IQC material. And what we mean by this is that if you're going to be doing 10 tests on 150 microliters, the machine might start to vary on the precision when you get really low into the dead volume. So use IQC first of all. Run that over and over and over at the levels that you're expecting to get the samples at, and check again to the levels that you're expecting, and make sure that you're not getting a variance at the low levels. And also, it's importantly that you can get those 10 tests from that sample volume. If you can't do it on IQC, don't waste your time with the capillary sample, which is obviously really, really precious. And then consider the different analytical variables that you can do during the testing. I like to build in as many variables as possible in my testing. So we'll do it on the same machine, but we'll do it at different times. So we won't always put them all on together and control that. I want a worst case scenario. We kind of treat it like we do a patient sample, as you should do with things like EQA, where you just run them and see what results you get. And we prefer to do that because that's like the worst case scenario. You may not want to do that. You may want to control it much more tightly. And as we've said, there are the statistics we've recommended. The two of them there was Bland-Altman and Passing-Bablock. So the validation and review. We've said that 40 pairs, 20 with justification, build in whole blood stability if it's appropriate. It might not be, But then when you've done all that, when you've got all your tests and you've done all the stats, look at the results you've got. And you may need to add in a couple more. There may be a variance on some. We've recommended that you do it in singlicate because, again, this is the worst case scenario. What you don't want to see is a lot of variation, because you're going to see that with real patient results. So if you are seeing a bigger variation, then it may not pass the statistical criteria that you've chosen. But when you do the review, ensure that your stability has been done and it's got a valid point of reference. Look at your rejection criteria and make sure there's not a downstream effect on the patients. Look at the numbers of insufficiency and hemolyze that you've got. And then that will need to be reviewed later on as well. So once you've done the project, we recommend that you do on a periodic time frame that you look at the patient results that you're getting. Now, there's two things here that we do. We look at capillary results and compare those to venous results. Now, we use a certain patient cohort. So my lab is generally well patients, so they marry quite nicely. But if you've got let's say a dialysis cohort that you're using this on, then look at the results, and you're doing, for instance, creatinine, enzymatic creatinine. Look at the results that you're getting from capillary, look at the ones that you're getting from venous, and they should marry up. If there's a positive or negative bias, you may need to relook at the validation, because what you should be seeing is those ranges should marry quite nicely. Or if you've noticed that your patient results are always wider than the ones that you've used for your verification, then the validation, then you might need to start looking at adding in more tests, more samples. That will enable you to build a data set that's relevant to your patient cohort. And that's kind of really, really, really important, that you revisit your validation on a constant basis. So the other thing as well is your failure rate. You need some way of being able to go back to people and say, this is the failure rate that we're seeing. Why are we seeing this? Is there something we can do in updating the IFU? It's a constant source. I think my lab's on about version 13 or 14 now on our IFU. And we made one initially that was quite wordy. There was very few pictures. And we found that people weren't reading that. So we added in more pictures. If you do too much pictures and don't explain things, there's a balance. And that will literally probably develop and be reliant on the patient cohort. Some people are happy to read everything. Some people need more pictures. Some people want online content, so on and so forth. So that will have to be reviewed on a regular basis as well, to try and get that rejection rate down. It may be that you may need to change methodology, analytical methodology. One good example, as I've said already, is creatinine. The Jaffa method isn't appropriate for postal samples. The enzymatic method is. It's much more robust. We use a slightly different ferritin assay to other people. It's immunoturbidimetric. It's very, very robust. So you might want to look at that as well to find out where your failures are happening and what you can do to mitigate that. Some of it will be education. Some of it may be service improvement, service changes. But you should always go back and review and add all these reviews into that initial change control before it gets signed off. And I think actually I've been a lot quicker than I expected to be. I expected to be about 40 minutes. So my apologies. I've covered most of that in about half an hour. We're on to questions, please.
Wonderful. Thank you so much, Tim. That was brilliant.
Nerves pushing me through really, really quickly.
Yeah, but it was all understandable all the way through. It was fascinating.
Yeah, all right, okay.
We have questions in the chat waiting for you. For anyone that hasn't asked their questions yet, Tim may look absolutely glorious, but he has years and years of experience there as well. So please, he's here. He can answer questions. Start piling them in while we go from the top. So Teresa has put a few in. Thank you, Teresa. First of all, a comment to say that the sender requires an IATA certification in the US. And then also asks, how is the data supplied, validated, and brought into the database and electronic health record?
Okay, so I work in a private lab, so that doesn't really, we don't have to really we do worry about that because we pass on patient information, obviously. But if you're looking at, so one of the things that we've done, and I'm just going to try and look at what we've done in our lab, is that we get HL7 requests, health language 7 input into the lab. those samples come in with us. They're obviously appropriately labeled. We book them in, and the results go back via HL7. Our system identifies that those patient results are from a capillary sample. So our system won't allow you to export the results without having a sample type in there. And the reason for that, obviously, is that if you're seeing a slight positive or negative bias, or with some things like finger prick, you might get the occasional error because they've got hand creams on there, or HRT, depending on what test you're doing. So you may get a flyer that's related to something that is on the finger. Now, you don't see that as much with upper arm technology. It's much better, but you don't know. You know, they may be looking at sex hormones, and they're on TRT or HRT, in which case they're obviously going to be incorrect. So it allows, having that sample matrices in the report allows you to look at those and identify them. Some people have queried whether or not you should actually have a different set of test codes for capillary, and that's a really good idea. Again, you can toggle between the two, but what I would recommend is that obviously when the results go out, they do go with that patient, that sample matrices on there, so that when it goes over to the patient record, it has that, the fact that it's been self-collected on there.
Brilliant. Thank you so much. I hope that answered your question, Teresa. We also have something that is the best thing ever to happen in comments. We have a debate. We have people helping each other. So Amy asked the following question, and Anne-Marie, you were amazing and already started answering it. Amy asked, once you have a validated capillary, as you have described here, if you switch methodology, for example, change from Roche chemistry to Abbott, do you just include capillary samples in your new method validation, or do you need to do another comparison study with paired capillary venous samples? And Anne-Marie has already answered with, because the chemistry between test developers may be different, it's best to do a comparison for each chemistry. Risk assessment would be required. Would you like to wade in on that discussion?
So there's a number of things. Yeah, I agree. I would redo it all again. So when we first started off, we were a Beckman lab. We were doing Beckman chemistry, Roche immunochemistry, and the reason for that was that the Roche uses a much smaller sample amount than other providers, and obviously we've got a very, very small sample amount, so therefore we've kind of moved all over to Roche. But when we first started, we started with Beckman, did all the chemistry on Beckman. But when we moved over to the Roche, we did it all again. I mean, I think it was something, when we did the Beckman ones, we did something like 70, 80 patient comparisons, because we were probably one of the first to do it, but there wasn't a lot of papers out there for all the assays we wanted, so we did quite a lot of work. When we moved over to the Roche, we did stick to the 30-40 mark, and we've added to that every year since then. When we've reviewed the patient results and gone, okay, we'll be seeing some low ones and some higher ones, and we've added to that database. But yeah, I would do it again.
Wonderful, thank you. We have another question here from Teresa. Do capillary samples need to be centrifuged? And if so, what is the general timeline from sample to processing?
So if it's self-collected and it's in the, in the home, for instance, it's probably going to be 2 or 3 days before it gets back to the lab. In the UK, we use Track 24. Depending on the time of year, next day, if they post it at the right time, next day is about 7% samples we get the next day. It goes up to about 98, 99% within 72 hours. So all of our stability work is done at 72 hours. We've done a lot more stability with venous samples as well over different temperature ranges, but when we do capillary work, we tend to do it at 3 days and usually at ambient temperature to start off with. So when you choose, if you think that capillary is appropriate for your patient cohort, you'll have to build in that stability. And if you've got a method, take for instance AST, very sensitive to hemolysis, then you're probably not going to get AST results because 20 or 30 of them are going to be hemolyzed. And if you need that assay, then capillary is probably not appropriate, unless of course you can't get results from those people normally, in which case a 30 or 40% failure rate might be appropriate for you. I mean, right now using the upper arm technology, we're seeing failure rates in the single digits for most of the analytes that we offer, but we don't offer potassium, for instance. There's absolutely no point. Calcium's another one we don't do on finger prick. So you've got to choose your markers to make sure they're robust enough.
Brilliant, brilliant. And you accidentally answered one of the questions further down. So Chris, Your question about what sort of sample failure rates do you see, please write again if that answered your question or not, because it was a very pragmatic decision on what you offer, and then you're getting down to single figures, aren't you? On the one.
So when we first started off with things like finger prick, some of the failure rates were double digits, 10, 15%, depending on the marker. Some markers are obviously very, very robust, and you'll find that out during your verification. Some markers aren't, in which case you'll need to decide whether or not you offer that and the reason behind it.
Brilliant. Brilliant. Moving on to the next one. Maros, I apologize if I pronounce your name incorrectly. Do you have any advice on best practices for validation of conversion factors serum to blood? This is especially relevant for dried blood samples.
Right. I'm going to quickly answer that one. Absolutely not. I've done hardly any work on dried blood spots. We do get asked for it occasionally, but we've focused on wet blood. I've done virtually no work on dried blood spots. There's a lot more to it than I've got the knowledge for. But I know that it's quite big in Europe. So I know that there's a group over at, I think, Gothenburg have done a lot of work on it. I think Utrecht, there's some great research coming out of Utrecht. So no, I don't is the answer to that question, I'm afraid.
Okay, short and sweet. Audrey has a question here. As capillary blood testing gains wider acceptance in routine clinical laboratories, how do you see this role evolving within clinical trials, and what barriers remain to broader adoption? And can I just pause a second to say thank you, because you submitted that beforehand as well. So thank you. It's been noted and very much appreciated.
So so the barriers are kind of like what we've seen in the clinical laboratory. You've got to choose the upper arm technology, the transdermal technology, has really altered this, the opportunities, the potential of this technology. They're still quite expensive, but that'll come down with more people using them and higher volumes. But that technology has allowed really good sample volumes, good sample quality. It removes the potential for things like milking the finger and getting hemolysis that way. So the upper arm technology has removed a lot of problems that we used to see with finger prick, but it is more expensive. For some clients, that will make a difference because getting hold of that sample is more important. And the other barriers are things like getting samples to the lab. Obviously, postal pathology has been out there for a long, long time, but temperature is obviously an issue. And with some of the pharma processes, that'll have to be tightly controlled. Now, I have seen little centrifuges that might be available. There is temperature controlled transport that I've seen in the US. So there's ways and means around it, but that obviously increases the cost. Choosing and proving that the analyte that you've gone for is very, very stable is probably the best thing you can do. And that would remove most of the barriers. If you're finding a bias, then it may not be appropriate.
Yeah, and also to the cost, it comes back to what is the need as well, because looking at Karen's case study in people with learning difficulties, if you have a trial population that perhaps is highly prevalent with autism or some other characteristics that mean it's difficult to get a sample from them another way, again, the need increases. Or from the clinical trial that PCSIG ran. And if you've got frequent PK samples and that you want them once a day, then you're more likely to get them this way. And people be happy with them. So it comes back to need as well, doesn't it?
And that, again, that starts off with when you do the validation, you look at the whole thing, you look at the whole patient pathway before you even start taking blood and doing the analysis. Make sure that I'm going to say business case, but make sure that the case for using capillary is really, really robust.
Yes. And you've thought through what people can achieve from the start when you building in your logistics.
Yeah.
Fabulous. We have a comment from Anne-Marie. Gosh, this is going to test my reading skills. Sometimes test manufacturers do not provide all matrices in their commercial review submission to NB or FDA for ease of approval, but will publish a technical note on the validation. This will not be included in the IFU for the test, but will be helpful in a risk assessment for verification versus validation. You just have to hunt for the publication from the test developer. Do you have any hints and tips to help people do that?
No, I don't. But I would still say that if it's not listed in the IFU, you still have to do a validation. However, more and more manufacturers are adding the capillary to their IFUs anyway. And certainly before we would start a project, we would do a publication search and try and find matrices that have been, evidence that it's been done before. If no one's ever done it before, then there might be a reason for that. However, there are lots and lots of papers out there now having proven that capillary and venous are comparable for a whole range of different analytes. So you should be able to find it, but you'll still have to do the validation, because in the UK at least, if it's not on the IFU, it's not CE or CA marked. For that purpose, you're using it off-label.
Thank you very much. We have a next one. Oh, Jenny, I am so sorry. Are the current capillary tube additives restricted to dry, for example, EDTA, etc., or are liquid stabilizers also acceptable? Are there differences in hemolysis prevention, efficacy between capillary and venous blood draw for dry additive stabilizer tubes?
So most of the ones are dry, as you say. I think if you're using wet ones, you probably want to look at your risk assessment as well, because this is going to go to a patient. And the reason why a lot of places went to dry was because there's a dilution effect possibly, and obviously if you've got a very small sample, that dilution effect is going to increase. So because the venous tubes are mainly dry, and because the capillary ones are mainly dry, then that's comparable. So that's ideal, really. Mixing between dry and wet, you're gonna add in a variable that you don't really want.
Okay, right. We have also another question here from Miranda. Would you be able to share the case study in learning difficulties? There you go. I was going to say it was on a previous webinar, and thank you for sharing that webinar. There is also a lovely video that was produced that I think we have housed maybe in a LinkedIn post, maybe on the website. I don't want to go hunting for it now, because if I do, everything will fall apart. But I'm sure we can share it after this. It's a beautiful video that shows the difference from parents and children's perspective that this made. It's honestly, it's amazing. Another one from Chris. What usable sample volume do you achieve with a finger prick sample? Thanks.
Right. Okay. So this is a good one. So we use different tubes. We use Griner for our kits. We also use BD. Obviously, the Griner ones are slightly bigger, in that the little label on the side is just slightly higher, and you can get like a mil or 0.8 microliters from one of those tubes. The BD ones tend to be more standardized, around the 500 or 600 microliters. Obviously, you spin it down, we're looking at half of that. So we've got a patient cohort that are extremely dynamic and focused on getting blood samples, and we will get 0.8 easily. And for us, 400 microliters of serum is venous levels. The Roche will do 30 odd tests on that sample, no problem. And likewise with BD tubes, we're getting 200-300 microliters from a tube. That's more than enough for that. So the thing about the finger prick is that it's reliant on the individual squeezing and milking. And although we get a lot of people that do it, they might use 2 or 3 needles, finger pricks rather. And eventually, if they do that on a regular basis, they're going to get quite fed up of it. And also, sometimes I've walked in and seen people do it, and it looks like there's been an accident where there's blood everywhere. People are trying to put blood on tubes and things. There's all over the tables and on this everywhere. So the upper arm device is kind of remove that variability. But BD is kind of used mostly for these devices. We get 300 microliters from an upper arm collection, and we've got some clients that are using it for 2 tubes. An EDTA and a serum and having no problem at all.
Brilliant. We have a question from Anne-Marie, I think relating to this topic we're talking about. So I'm going to jump to that one next. It says, is this also true of BD mini draw device?
So yeah, I have used a mini draw, and the mini draw is really, really good. There's a couple of, let's say, foibles about it that you have to, get the finger fit right. But a lot of it's done for you. And yeah, and those, they again, I think they're 600 microliter tubes. They use a slightly different tube. it's an upside down tube, so it pops out quite nicely. But then the processing is a lot easier than a normal tube for that reason, for the lab. But yeah, you're going to get 600 microliters, no problem with that device.
Brilliant. And Zora says, is there any data on comparable cost of capillary testing compared with the cost of repeated attempts at standard failed blood test attempts on, for example, patients with learning difficulty?
So I can't talk about the patient cohort, but there is a few papers. I'm trying to think. I think it's Levenko, or there's Martel et al. As well, that have looked at dry blood spot costs in patients. So if you think about the cost of a phlebotomy collection compared to a capillary collection, you're looking, if you add it all up, it's going to be variable depending on what level of person you use. But if you took like a nurse, sort of like a GP nurse, you're going to be looking at something like £30,000 for the time, £10,000 for the on costs, probably £7,000 or £8,000 for the clinic costs, and it kind of comes down to around about £4 per collection. When you add in all the other things, it comes to around about £8 or £9. But of course, that depends on where you are. If you've got a clinic that's overlooking the House of Parliament, it's likely to be a lot more. If you've got a clinic somewhere up north on a business park, it's going to be a bit cheaper. If you can get someone that's cheaper to do it, so on and so forth. So it is really comes down to the different variables that you include in that collection. And then when you look at the capillary side, the device and the postage is actually the main cost parts, and you're going to be looking at £12 or £13. So overall, it would appear for most patient cohorts that phlebotomy is slightly cheaper than capillary blood testing. But then you've got to look into things like the different patient cohort. If there's a learning disabilities, do you have to do a certain amount of extra work to familiarize them with the collection process? That might be extremely costly. But when you start adding in things like the societal cost, that's when it changes. So when you start looking at the time it takes for someone to go to a GP or an outpatient, the average is around about half an hour journey there, half an hour journey back, plus 10 minutes on site. So you're looking at basically an hour and 10 minutes. So when you start looking at the societal cost, phlebotomy is a lot more expensive than capillary. And there's a few people looked at studies and have shown that it is more expensive when you include societal cost, but it would depend on a number of variables: who's collecting the blood, how efficient the clinic is, and the patient cohort that you've got.
Brilliant, thank you. We've got lots of people wanting the case study for Karen's case study for learning disabilities. Rav, you are a superstar. If you look in chat, Rav has shared the link to the previous webinar where Karen was here talking through precisely what happened and all of the data from the case study. And the video, thank you for also sharing that, is on YouTube. So just look in chat and you'll be able to pull the information out of there. And we have a question from Michael. Is there a list of good upper arm devices available in the UK market and ones that are approved for use in adults and children? We're looking for something, it's changed, beyond Tasso.
Right. So there used to be the TAP, but the TAP has kind of gone off the market now, unfortunately, because it's been bought out by another American company, and they're looking at reevaluating the design of the device. So right now, the only one that I know of for there's two actually. So there's one called, I think, Presley Health released one, which is CE marked, but that uses an upper arm device that actually takes a blood tube, and it's not CE marked as far as I'm aware for children. In fact, I don't think any of these are actually CE marked for children. I think the Tasso is. So there's another one coming to market soon from Let's Get Checked, the Impress device, which we've used. It's very, very good. At the moment, I think probably the best option you have is the Tasso. It's been used lots and lots and lots, and it's very, very popular. If you can get hold of the TAP 2, then fantastic. I know that people have been trying to get hold of the remaining stock because it is very, very good, especially for children, because it is more or less painless. So the Impress device should be out, I think, August, September this year. That's very good, but that uses 2 BD finger prick sticks built into the upper arm device. So it leaves 2 little holes, obviously like a finger prick does, but it doesn't leave a large cut, or a lancet cut like the Tasso can do. So that's very, very good as well. And I think by the end of the year, there's going to be several more on the market.
Yeah. And thanks, Anne-Marie. That backs up what Tim said, that Tasso has information on their website for use for small children. I've also included in the chat on the PCSIG website, if you look under the tab Technologies, there's a link of different technologies for samplers and everything else. I'm not convinced it tells you particularly whether the CE marks are in the UK, Europe, etc., but there are people that know more about this than I do, and the links
at least that gives a list of devices out there that's available, and then you can contact the manufacturers and see and ask them the questions.
Exactly. Another one from Michael: if you change collection device, do you need to redo your study? I think it's similar to one we had earlier.
Yeah, so this is a really interesting one. So I've had lots of theoretical discussions on this with lots of other people. So we've said no, depending on the collection technology and the tube. So we've taken the sort of like the angle that if it's the tube, there's still capillary, but that would depend on a number of different variables, how many you've done, and I suppose the tube that's in use, if that changes, the plastics, things like that. So what we've done in the past, we haven't done a full verification. We've done a much smaller one just to check, to make sure that that there's been no changes. And if I'm honest with you, when you move over device, you're going to do some work anyway. You're bound to, because if you're going to change device, you want to make sure that you get the right amount. You're going to want to get the patient buy-in. So you're going to end up doing getting some blood samples anyway. And I would just plan that technical file part in with my validation again and add those samples in. So we had a discussion not so long ago in summary about it. We said we probably wouldn't need to, but when we've moved device, we've always tested it to make sure it's as good, if not better, than the previous one. And to do that, you get blood samples, and then we've included that in our thing anyway.
Wonderful, thank you. And I think we have the perfect comment to end the questions on from Sam.
Please leave.
Saying thank you for a fantastic webinar. They now just need to infuse their local laboratories. They work in LD services as a specialist LD physician and have so many complex patients for whom upper arm method will be transformational for their healthcare. So thank you. And thank you, Tim. Thank you for everybody that has put questions in the chat. That has been amazing. Before we leave, I would just like to move on to the next slide, just so that people can, if you are not already a member of the Patient-Centric Sampling Interest Group, we're a friendly group who try and help to work together to solve some of the problems and try and make this easy for people. You can go to the website, which is on that QR code that I've been posting links to useful things from, or join the LinkedIn group and you get posts, including things like the video that Karen put out and stuff. So please take a note of those as we go past. And another exciting news item that is very relevant for Tim is the birth of the Journal of Patient Centricity. I don't know whether you want to say anything about this, Tim?
Yeah, so it's a new journal. It's basically been designed for this purpose. So for patient-centric testing, it's not just, I mean, obviously I'm keen on the capillary side, but it's not just about that. It's about things like wearables, point of care, different collection methodologies. Doesn't have to be blood, but it's all about how we bring the patient into the testing of the samples, really, and making it much better for the patient. So I think our first edition's just being released. There's a few papers in there on capillary stability. There's been more added in, and over the next few months there'll be more and more and more. So please, either if you've got an interest, have a look at it. If you've got any paper ideas, you want to put a paper in, please do. What we're trying to do is have a central place for all patient-centric testing, so that it's easy for health professionals to go to one place and look up what they need. So it's much easier to find.
Brilliant, thank you. And I think it's worthwhile also mentioning there is quite a diverse editorial board that includes patients as well.
Yeah.
So thank you again to our partners and sponsors. We really appreciate all the efforts and contributions that you do. And this is just the statement at the end while I say we're going to be taking a short break over the summer while everyone goes on holiday, and we will see you again just after the summer holidays in September, October time. So thank you, Tim. Thank you, everybody, for listening and watching and all the amazing comments. I hope this really helped you, and have a lovely evening.
Thank you very much. Bye-bye.